The real RNA-seq problem isn't sequencing.
RNA sequencing generates large amounts of data, but when input quality is compromised, more data only amplifies the problem. Most RNA-seq failures start long before sequencing begins. In the real world, RNA rarely arrives perfect. Clinical samples, archived tissue, FFPE blocks. By the time RNA reaches the lab, it's already fragmented, degraded, and unpredictable. And when degraded RNA enters traditional library prep workflows, researchers face inconsistent libraries, sequencing noise, and unreliable results. Because successful RNA sequencing starts with better tolerance for lower quality RNA. Introducing truCOVER Total RNA Library Prep from Covaris, a fast, streamlined RNA-seq workflow engineered for real-world RNA samples, even for degraded FFPE RNA. At the heart of truCOVER is adaptive focused acoustics technology. AFA uses controlled acoustic energy to extract and fragment RNA precisely and reproducibly, creating consistent whole transcriptome coverage while protecting RNA integrity. The result? RNA libraries that are highly reproducible, strand-specific, and evenly covered across the transcriptome. Even from FFPE RNA with DV₂₀₀ as low as 30%. truCOVER Total RNA Library Prep supports accurate detection of gene fusions found in nearly 20% of human cancers, and enables discovery of alternative splicing events that reveal deeper layers of gene regulation. The entire workflow completes in just 4 hours. Fewer steps, less hands-on time, automation ready. When combined with Covaris truExtract FFPE extraction, researchers gain a complete RNA-seq preparation workflow, from extraction to sequencing-ready libraries. From a tiny piece of tissue to discoveries that change lives, every RNA-seq result depends on preparation done right. Covaris truCOVER Total RNA Library Prep. Better preparation, better libraries, better sequencing.